Okay, so this last video is going to be relatively short, where we're going to get into membrane potential. So we're going to define what that even means. Then we're going to look at a few things that contribute to establishing this membrane potential, especially the negative inside of the cell, and then talk about how this can change and how this contributes to impulses in the neurons. So the cells of our bodies, right, they're all regulated, and we've been talking about how they get regulated a few different ways so far. I mean, one of the big things we've already talked about is how we regulate the solute concentration inside and outside of the cell. And we talked all about the transport, right, that handled regulating solute concentration and osmolality. But we also regulate the charge inside and outside of the cell, in that cells want to be more negative on the inside compared to the outside. So we want to have fewer positive ions inside the cell, more positive ions on the outside. This is basically so that we know we have a charge distribution on each side. Now, why do we have the charge difference? A lot of it comes down to this permeability of the different ions. So things like potassium, sodium, calcium, chlorine ions, right? Those will help contribute to the charge, right, to the membrane potential, as well as the action of the sodium potassium pumps, right? So pumping out sodium, pumping in potassium. And then there are a lot of negatively charged molecules and ions inside the cell as well. So something like DNA is negatively charged. So we see a lot of that, right? A lot of that going on within the cell, that we're trying to maintain this negative charge. Now, the difference in the charge between the inside and the outside of the cell is called the membrane potential, right? So you're going to see membrane potential come back. You should be able to define it, right? Just the difference in charges from each side of the membrane. And we measure this in millivolts, so really small, right? So you'll see MV, that's millivolts. Now, in a resting state, or the resting potential, right, of a cell, right, is going to be when we're in a non-activated state, right? Like rest, right? If you're resting, you're not doing anything active, right? So the potential difference that we see when the cell's just not doing anything, especially if we're talking about a neuron, neuron is not firing a signal, it's not doing anything. The difference between the inside and outside of the cell's charge is called the resting potential. And this usually comes down to a couple things. It comes down to the ratio of the concentration of ions, in particular potassium and sodium. And when we talk about the neuron, we're going to really talk a lot about the movement of potassium and sodium ions. And then also it comes down to the permeability for each of these ions at any given time when we look at the transporters in the membrane. So in general, we have a ton of sodium found outside the cell. We have some potassium inside the cell. And so since we have more positively charged ions outside compared to the inside, it's a net negative charge. And generally speaking, when we're talking about like a neuron, its resting potential, the charge is a negative 70 millivolts. And we'll get into the details of like what happens and how do we fire an action potential a little bit later. But this is really key. We want to have this idea of resting potential and understand what a membrane potential is now, because we will eventually be talking about neurons and muscle cells and how they fire and how they do their job, and they're reliant on these changes in charges. They're reliant on these ions moving in or out, right, in various areas to allow for that action to occur. So like I said, we said the negative 70. Most cells are going to be somewhere around that 70, negative 65 to negative 85. So we're usually within that range. That would be homeostasis, right? Some cells, like the neurons and muscle cells, they're going to go through a process where they'll actually shift from negative to positive. So for example, like if we are a neuron, we'll shift from negative 70 up toward, through negative 55 up to positive 35 millivolts. And that's for the depolarization and repolarization process that allows for that impulse to be sent, right? So things like neurons and muscle cells, they're actually going to have moments of well, I guess we'd say instability in terms of that potential, right? We change that charge to allow for us to do that activity. But most of their cells are going to stay in that negative 65 to negative 85 range. So I just mentioned the words depolarization and repolarization. So let's go ahead and define those. So impulse propagation, basically the start or the continuation of an impulse, right? This is going to be because the membrane permeability to sodium changes, allowing sodium to flood in. And if we have sodium flood in, our charge goes from something like negative 70 up towards maybe negative 55, up towards 0. And if we're something like a neuron, we can go up to positive 35. So sodium enters in. This changes this changes the charge inside the cell. So we say that we've depolarized the cell membrane because we've flipped it, right? So we've gone from the more negative on the inside, more positive on the outside, to now we're positive on the inside. So we've changed the charge. And that'll be used, again, like I said, for like an action potential of a neuron. Now, we don't stay in that state forever. We do go back, and that's where we go through a process called repolarization, which is basically to get us back to our resting potential, back to our starting point. And so this is where we are going to pump that sodium out, pump potassium back in. Remember, it's a 3-2 ratio, so 3 sodiums pushed out, 2 potassiums back in. So that means for every time we use our sodium-potassium pump, we're losing 1 positively charged ion in that process. So that's how we're coming back down, right? Back to that negative charge again, because for every 2 we're bringing in, we're kicking 3 out. So we're losing 1 positively charged ion every round we go through that sodium-potassium pump. And that gets us back to our resting potential voltage, and then we can restart again. Again, we're not getting into the details, so don't worry about understanding how a neuron's action potential functions just yet, but you should be familiar with the terminology here. That's the one thing in this little short video I care about, is terminology. Understand what membrane potential is, understand what resting potential is, and then understand the concept of depolarization and repolarization. If you have those things down, then I'm pretty much happy with what we cover in this little section here.